In this presentation, we're going to be looking at Thomas Hunt Morgan's work and his experiments looking at linked genes in fruit flies. Now, Thomas Hunt Morgan actually created specific recessive characteristics in the fruit flies by exposing them to different mutating chemicals and X-rays. And he used these mutant fruit flies with these recessive genes in breeding experiments with the normal wild type flies that have the dominant genes. Now, through his breeding experiments, he did many breeding experiments, and he was able to actually map the chromosomes in Drosophila and actually show the position of each of these genes. Now, because he created these mutations, he actually named the genes after the mutation rather than actually the wild type example. So, for instance, we're going to be looking at two different genes on the same chromosome. This is chromosome 2. We're going to be looking at the inheritance of this, which is black body, and then the wild type version is the grey body, so that's the normal dominant characteristic. And we're going to be looking at vestigial wings, which are basically really short, not really functioning wings, which is the recessive characteristic, and then the normal long wings, which is the wild type characteristic. So these two genes, we're going to be looking at how they're inherited. Because he created all these recessive characteristics, he actually named the genes after the recessive characteristic. And in this experiment, we're going to be looking at a heterozygous fly and a homozygous recessive fly for these two particular genes. So you can see this one here is our homozygous recessive fly, and it's got two recessive genes, one for vestigial wings and for black body, both recessive characteristics. Whereas the other fly, this one here, is heterozygous. And you can see the notation here. Instead of using a capital letter, they use VG+. So it's not vestigial wings, it's normal wings, but it also carries the vestigial wing allele as well. And this one here, BL+, is dominant characteristic, so that will be the sort of greyish yellowish body, whereas the recessive one here is the one for black body. So we're going to be looking at this particular cross now. So a linked gene test cross. So a heterozygous grey fly with normal wings was crossed with a black fly with vestigial wings, and I've shown you the notation here. So we'll do this particular cross, and I'll show you how you can do it. So with our parents, there's our parents. We're going to show the heterozygous, which is this, and this, and we're crossing it with the homozygous recessive, so that's just this. And I've drawn the alleles, the chromosomes horizontally here, so you can see the pairs of alleles. And then in producing the gametes, well, this one here only produces one kind of gamete. So it can only produce a gamete like this. There's no other kind of gamete that this one can produce because it's homozygous for both of the genes. This one here is heterozygous for both. But because of the linkage, you're going to get lots of these ones. So you're going to get lots of this. And you're also going to get lots of this because they're inherited together. But you're also going to get a little bit of crossing over taking place. And here's showing a chiasmata forming. And then we swap over the alleles. And so these will produce these Okay, so this one, you've got 4 different gametes being produced, and we'll put them in circles, but you'll get far fewer of these because these have been formed through crossing over. Okay, so we can put this now into a Punnett square, and because it's a test cross, we just need a skinny Punnett. So this is our offspring, and we've got just a 1x4 Punnett square, like this, and we can put this one here. So this is our recessive one, and then we've got the 2 dominant, and we've got the 2 recessive, and then we've got the dominant with the recessive and the recessive with the dominant there. And if we fill that in, I'll just put the lines in first just to make it easier. And I'll put in all of the and then I'll put in the other one. And so we can see here what our phenotypes will be. So let's put our phenotypes. And the phenotypes will be grey and normal wing. And then this one will also be grey, but we've got the vestigial wing. This one will be black with the vestigial wing, with the normal wing. And finally we've got the black with the vestigial wing. Now if they weren't linked, you'd expect a 1:1:1:1 ratio in the offspring, but because we have far fewer of these gametes, we don't get a 1:1 ratio, and this is what Thomas Hunt Morgan found. And in fact, he was expecting a 1:1:1:1 ratio, but in fact he got 5:1:1:1. So a lot more of the wild type characteristics, so the greyish body and the normal wings, and the black body and the vestigial wings. And these two, obviously far fewer, because they are produced through crossing over, and you get fewer of those gametes that are needed to form those offspring.